Hello everyone, I am Dr. Sirajul Faizi. I welcome you in my YouTube channel. Today in this video, I am going to talk about classification of enzymes. So classification of enzymes, a systematic classification of enzymes has been developed by International Enzyme Commissions. And this classification is based on the type of reaction catalyzed by enzymes. There are 6 major classes. Each class is further divided into subclasses, subclasses, and so on to describe the huge number of different enzymes catalyzed reactions. So, enzyme class, reaction type, and example. Oxidoreductase, reductions oxidation. Lactate dehydrogenase. Transferase, move chemical groups. Hexokinase. Hydrolysis, bond cleavage with transfer of functional group of water. Lysozyme. Lysases, non-hydrolytic bond cleavage. Fumarase. Isomerases, intramolecular group transfer, isomerization, triose phosphate and isomerase. Ligases, synthesis of new covalent bond between substrates using ATP hydrolysis, RNA polymerase. Now friend, mechanism of enzymes actions. Mechanism of enzyme action, the catalytic efficiency of enzymes is explained by two perspectives, thermodynamic change and process at the active site. Thermodynamic changes means what? All chemical reactions have energy barrier between reactants and product. The difference in transition state and substrate is called activational barrier. You can see in this picture transition state uncatalyzed, transition state catalyzed, and their free energy, and this graph is free energy versus progress of reactions. Now friends, thermodynamic changes. Only few substances cross the activation barrier and change its product. That is why rate of uncatalyzed reaction is much slow. Enzymes provide an alternate pathway for conservation of substrate into product. Enzyme accelerate reaction rate by forming translation state have a low activation energy. Hence the reaction rate is increased many folds in the presence of enzymes. My friends, the last point, the total energy of the system remains the same and the equilibrium state is not disturbed. Now, thermodynamics change overview. You can see without enzymes and with enzymes. So glucose, galactose, and here glucose, lactose with enzymes. Activation energy with enzyme, net energy release. Activation energy without enzymes. So, process at the active site. Covalent catalysis, acid-base catalysis, catalysis by strain, catalysis by proximity. So covalent catalysis. Enzymes form covalent linkages with substrate, forming transient enzyme substrate complex with very low activation energy. Enzymes release unaltered after completion of reactions. You can see the substrate, enzymes, transition state, and then you can see the products. So acid-base catalysis, mostly undertaken by oxidoreductase enzymes. Mostly at the active site, histidine is present which act as both proton donor and proton acceptor. Catalysis by proximity. It is catalysis molecules must come in bond forming distance. When enzyme binds, the region of high substrate concentration is produced at the active site. This will orient substrate molecules, especially in position ideal for them. So, catalysis by bond strings, most undertaken by lysases. The enzyme substrate binding cause reorientation of the structure of site due to strain conditions. The transition state is required and their bond is unstable and eventually broken. In this way, bond between substrate is broken and converted to products. You can see in this pictorial diagram how substrate and active site, they are making enzyme substrate complex. Then you can see the enzyme product complex and their products. My friends, lock and key models proposed by Emil Fischer in 1894. Lock and key hypothesis assumes the active site of an enzyme are rigid in its shape. There is no change in the active site before and after chemical reactions. So, induced fit models. More recent studies have revealed that the process is much more likely to involve in induced fit model proposed by Daniel Koch-Land in 1958. According to this, exposure of an enzyme to substrate causes a change in enzymes which cause the active site to change its shape to allow enzyme and substrate to bind. So induced fit model, you can see the substrate, you can see the active site. So enzyme kinetics. My friends, I will discuss from here in our next video. If you like this video, then subscribe my channel, and thanks for watching.